Okay. So yeah, last week we learned until we learned until carbohydrate, right? Carbohydrate and our lipid. Okay. Yeah, so right now let us continue with our chap subtopic 2.3, which is our protein. Okay, so today we'll be looking in protein. So for protein, right, okay, they are actually a quite major, they are actually a quite major subtopic lah inside your biological molecules. Okay, so there are a few things that you need to know. Okay, first one is what is your amino acid structure and the peptide bond. Second one is our 4 levels of your protein structure. So this one is very important. This one, please put the stars here. And for this subtopic, you must know the 4 levels of protein structure. Okay, because majority of question comes out from here. Okay, and third part, we will have protein shape. Okay, third part, we will have our protein shape. So for our protein shape is actually related to your 4 levels of your protein structure. Okay, this is actually our protein shape. And number 4, we will have different types of globular and fibrous protein. This is we call it types of protein. Okay, this one we call it types of protein. And then fifth part is actually our hemoglobin. Okay, fifth part is actually our hemoglobin. So hemoglobin is actually how you form your red blood cells component. And the last one is our collagen. Okay, so this is the outlines of this chapter lah. Okay, so yeah, first one. Okay, proteins, they are actually polymers. Okay, for proteins, they are polymers. While for protein is polymers, while for monomers, they are called amino acids. Okay, monomers, they are called amino acids. So proteins, they are actually extremely important in cells because they form all of the following enzymes, hormones, cell membrane protein, all those immunoglobulin, example, hemoglobin, keratin, and collagen, or even myosin. So you can know that all of your structure in our body mainly composed of protein structure. Okay, mainly composed of protein structure. Okay, so this is actually the proteins, while the second one is our amino acid. Okay, second one is our amino acid. So amino acid is the smallest type of your protein. So you roughly have 20 amino acids found in your body lah. And the general structure you need to know. So you will have one amino group, which is actually basic. Okay, or you want to call it alkaline, also can. They're actually basic. And you got one R group. Okay, R group means variable side chain. Okay, the R group means variable side chain. Variable side chain means that the chain the chain is composed of different properties. Like example, if your side chain is acidic, they will cause your whole amino acid to become acidic. If your R group is basic, okay, if your R group is basic, so you will cause your amino acid to become alkaline. Example, okay. So this is the R group. That one we will see later. And you got also carboxyl group, COOH. Okay, COOH in your chemistry we call it carboxylic acid. Okay, COOH we call it carboxylic acid. So carboxylic acid, or we call it carboxyl group, they are actually acidic. Okay, so this is your amino acid structure. That's why the amino refers to amino group. The acid refers to carboxylic acid group, or we call it carboxyl group. So this is their structure. Okay, the general structure you need to know. And you need to remember the structure of this one, because sometimes they'll ask you. So you need to remember the structure, the general structure. This is called general. Means that the R group, you didn't list out the specific one, you just write the general one.
Okay.
Okay. Then, okay. After that, okay, let us look at this part. Okay, we also got peptide bond. Okay, peptide bond. So for peptide bond, okay, peptide bond is same as the one. Okay, remember not? Okay, we directly use this structure to explain. So same thing. Last time we learned about glucose and glucose. You will form glycosidic bond. Okay, glycosidic bond. And last time lipid, you will form ester bond. Between who? Between your alcohol, OH, along with your carboxylic acid, COOH, which is the glycerol and fatty acid. Okay, so this is the third bond that we need to know. We call it peptide bond. So peptide bond refers to, okay, peptide bond refers to your how to say, refers to your protein. So same thing, condensation, you throw out one water, and hydrolysis, you use water to break down. So when I combine two amino acid, okay, when I combine two amino acid, you combine OH and H together and you form a water. Okay, you will form a water. So after you form a water, you combine them, the structure between C double bond O and NH. So C double bond O and H, this structure, we call it, in chemistry you will learn it called amide. Okay, you will learn it called amide. Okay, so this is the dipeptide. Okay, so dipeptide, between these, we call it peptide bond. And peptide bond is covalent. Why? Because all of them they don't have any metal in it. They don't form any ionic bond, ionic molecule. Okay, so this is your peptide bond. Okay, so for this part, is it okay?
Yeah.
Yeah, can okay. Yeah, it's like similar that similar like your carbohydrate,
Yeah.
your lipid.
Yeah.
Yeah, yeah. So if this part okay, I directly skip. Okay, this is the main part for today lah. Okay, this one you put 2 stars. Okay, to be simple, you got 4 types of structure. Okay, 4 types of protein structure. So first one is primary structure. So remember the keyword primary structure. We talk about the sequence of amino acids. This one must write down. Okay, during your exam, if they ask you, you need to have these words called sequence of amino acids. Okay, sequence of amino acid bonded by covalent peptide bond. Okay, and your DNA of cells determine the primary structure. So what is the sequence of amino acid? You just need to know. Sequence of amino acid is like the chain. You see all the chain here? The chain is called primary structure. Okay, so primary structure, just need to remember one word called sequence of amino acid. Okay, so this is the first one. Okay, first one. First one, very easy one. It's just the normal amino acids. Okay, form a long polymer called peptide chain, or you want to say protein also can. Okay, so this is your first one. While secondary is the second level. Okay, second level, you got a keyword called alpha helix coil and beta pleated sheet. Okay, alpha helix. Okay, this is actually a coil lah, although they didn't mention here. And this one is a beta pleated sheet. Okay, so how do your alpha helix shape form? So your alpha helix shape form when your hydrogen bonds form between every 4th peptide bond. Okay, later I'll show you. While beta pleated sheet forms when your protein, okay, your protein folds so that two parts of the polypeptide chain is parallel. So you see, one is form a coil, okay, one is form a coil, another one is form parallel shape one, okay. So this is the secondary part. Okay, we directly see this one first here. Yeah, so you see alpha helix is actually a coil, okay, while beta pleated sheet, you can see they are actually folded. Yeah, you can use a word called folded to describe the beta pleated sheet. Okay, normally we use folded. Okay, they are actually called folded, and this one is a coil. So you see how we form a coil. You see, it's like a 1, 2, 3, and fourth. Just now we say fourth peptide bond, right? So you can indicate as fourth carbon. So you see 1, 2, 3, 4. Every 4. There also got 1, 2. Okay, 1, 2, 3, 4. Okay, but this one just do as learn as reference can already lah. Okay, no need to like draw out. Okay, they will ask for drawing. Okay, so this is actually our beta pleated sheet and also our alpha helix. So you can see hydrogen bond here is like a connected to every fourth peptide bond, while beta pleated sheet is like, you see, they say parallel. In parallel in your mathematics means that both have the same length, have the same direction, have the same spacings lah. Okay, so we call it parallel. Okay, so you see, they are here refers to your parallel part. Okay, all these refers to parallel, and this is every fourth peptide bond they will form. Okay, so this is the structure. Okay, so for the secondary structure, is it okay?
Yeah, it's okay.
Yeah, okay. And most of your fibrous protein, they will have secondary structure. Like example, collagen. Yeah, collagen is provide elasticity to your skin. Okay, or they can be found in your bones or anything. Okay. And then keratin. Okay, what is keratin? Keratin is where the amino acids form in your nails and your hairs. That's why when you buy the shampoo, those things, right, they will say good for keratin production or anything. Okay, you will saw these words. For collagen is like for your skin lah. Like example, those cosmetic or anything, they will like say collagen enhancement example. Okay, so and where the food, some of the food you eat also high in collagen. Okay, yeah, so this is the fibrous proteins. And secondary structure only relates to hydrogen bond forming between amino acids and carboxyl group. Okay, so to be simple, it's like the primary structure. Okay, primary structure, you directly form peptide bond and you form a long chain. Okay, and then secondary structure is all about hydrogen bonds. Okay, hydrogen bonds. Right now, okay, let us look at tertiary. Okay, so tertiary structure, okay, you will undergoes further conformational change. Okay, you will form further conformational change. Okay, what does it mean by conformational? So conformational change means that the change in your structure. Okay, change in your structure and leads to additional bond. Okay, leads to additional bonds forming between your R group. Okay, forming between your R group, which is actually our side chain. Okay, which is actually referring to our side chain. So the side chain, okay, the additional bond here, you got 4 types. Let me discuss with you. Just now, secondary structure, we learned we only got hydrogen bond. Okay, we only got hydrogen bond. But tertiary structure special is what? You got 4 additional bonds. Okay, this one take note. Sometimes MCQ will ask. Sometimes your structure they will ask. Okay, sometimes MCQ they will ask. Sometimes structure they will ask. So first one, of course, you will have hydrogen bonds between who? Between your R groups. Okay, between your R groups. While second one, we will have disulfide bond. Okay, disulfide bond. So disulfide, they only occurs between cysteine amino acids and sometimes referred to as disulfide bridge. Okay, disulfide bridge you will look as here, CSSC. Like example, this is one R group and this is one R group. Disulfide means that they will got two sulfur to form something called sulfur. Okay, you will learn in advanced in another part called sulfur cross-link structure. Okay, to prevent sliding over each other. So normally we will have this structure. Okay, so this is disulfide. While third part is actually our ionic. So ionic charge, ionic bonds, I think you are familiar. You learn chemical bonding, right? So it's a metal and non-metal, not metal and non-metal part. And you also got weak hydrophobic interactions between who? Between your non-polar R groups. Okay, between your non-polar R groups means that non-polar means that hydrophobic group. Okay, because non-polar is for hydrophobic, polar is for hydrophilic. Okay, so this is the tertiary structure. And we also got tertiary diagram. So you see, primary is this one, long chain sequence of amino acid. Okay, let us do some summary here. Primary is your sequence of amino acid. Secondary, we got what? Secondary, we got alpha helix and beta pleated sheet. So these terms need to memorize lah, okay? Because they got a specific term for specific part. And tertiary, you can observe that they are getting more complex already. They are having more coil, more complex structure. Okay, and the distribution, let me show you. So H and O, you refer the key below, the table below. So you see, this is called hydrogen bond. Okay, refers to this one. Ionic bond is between positive charge and negative charge. Okay, because metal, okay, we in chemistry we learn ionic bond normally refers to metal, right? So metal, they will donate their electron to form positive charge. So ionic bond, they involve positive charge also. And here is negative charge. So your ionic bond, they will bonded with negative charge to form ionic bond. Okay, and disulfide, you see, I got sulfur crosslink. I got two sulfur in between your CH2. And the orange color part is actually our weak hydrophobic interaction. So you see, weak hydrophobic interaction normally is like a hydrocarbon. Okay, you can remember as hydrocarbon thing because they don't produce any charge and they are mostly covalent. Okay, they are mostly covalent and they are hydrophobic also. Or sometimes we refer them as non-polar. So you see, this is how the terms related in your chemistry, in your chemistry or in your bio. So this is the first tree structure. So first tree structure you can remember as you can write formation of bonds between R groups. Okay, or then you can write a conformational change lah. Okay, so this is the third structure. So
Okay.
this one is okay? Can
Yeah.
Yeah, okay, so if this part okay, let me show you. Okay, let me show you this one. Quaternary structure. Okay, quaternary structure, you occurs in protein that have more than one polypeptide chain. Okay, and one of the example in A levels we use is called hemoglobin. Okay, so hemoglobin you will learn in transportation, that chapter is actually involved in our component of red blood cells. Okay, they help you to transfer and deliver oxygen, carbon dioxide in and out of the body cells, okay, for respiration purpose. And each polypeptide chain in the quaternary structure, they are referred as a subunit of a protein. Okay, so you see, quaternary structure, you will have alpha subunit, beta subunit, beta subunit here, and alpha subunit here. So you see, they are like actually intercross. Okay, they are intercross at different position. Okay, they are opposite to each other, I would say. Okay, and since you got alpha globin, here you got beta globin. So these four structure together will form hemoglobin. And special part is that hemoglobin, each globin, okay, each globin, they carries one oxygen molecule. So to be simple, one hemoglobin, they can carries four oxygen molecules, and one hemoglobin, they can carry 8 oxygen atoms. And we got millions or even billions of red blood cells is traveling in our body. So we are constantly provided the oxygen by our red blood cell. And inside here, you got one heme group. Okay, heme group later you will see lah. So heme group normally they involve the iron Fe. Okay, iron Fe structure. That's why you heard they say like some of the food is high in iron, right? Examples, your beef. Okay, your beef meat or your mutton meat. Okay, it highly consists of iron. So it is good for those with anemia lah. Why? Because iron is needed in our body to produce much more blood cells. Okay, so this is quaternary structure. So we got 4 lah, okay? So this is the summary of bonds in your protein tables. So let us look through primary, secondary, tertiary. All of them got peptide bond. Okay, but secondary and tertiary only, they got hydrogen bonds. Okay, and the remaining one, tertiary, they got disulfide, ionic, and hydrophobic. So you can remember one thing is your tertiary structure almost have all the bonds, while primary structure only involves peptide bonds. They don't have any additional bonds here. So directly cross them lah. Okay, or you can like directly cross them, and here directly cross them. And secondary structure, they got peptide bond and hydrogen bond. Okay. Yeah, so this is the first part. Okay,
Yeah.
okay. So after that, let me find this one. The second one. Okay. Okay. So for proteins lah, let me see. Okay, they further discuss with you the interactions and their shape. They further discuss interaction and shape. So for this one, a polypeptide chain, they will fold differently. Okay, they will fold differently due to interactions, okay, between your R groups. So R groups is this one. These are different R groups here. So here you got totally 4 R groups. Okay, or to be simple, each of the part here, anything with C and H, normally we call it R. Okay, in your chemistry. That one you will learn in chemistry lah. Furthermore. Okay, and then okay, let me discuss here first. So 3-dimensional configuration that forms is called tertiary structure. So starting from tertiary and quaternary, these two, okay, these two, they got 3D shape. Okay, these two, they got 3D shape. Okay, 3D means 3-dimensional. Okay, why? Because they are complex structure. Okay, and we got 4 bonds as stated here. Just now we already go through. Okay, so let us look one by one. So disulfide disulfide bond, they are actually a type of strong covalent bonds that forms between two cysteine R groups. Okay, for this one cysteine R group, they are actually called cysteine is actually a type of amino acid. So these two cysteine amino acids are groups. How you link them together means that how that you form these two cysteine and join them together. So you use something called disulfide bonds, which is by connecting two sulfur atoms to each of their carbon on the cysteine. That's why you have CSSC. Okay, so this is disulfide bond. And normally we call them disulfide bridge lah. Why? Because you link them together. Okay, so we call it a bridge. And then they can be broken down by reduction. So this one you learn in chemistry called oxidation and reduction. Okay, we call it redox. Okay, and then we also got disulfide bonds, which are common in proteins secreted from cells called insulin. So tertiary structure lah. Okay, tertiary structure of protein. Normally we will produce a lot of hormone, such as your insulin. Okay, we will produce many types of hormone, okay, such as your insulin. Okay, and then second one, okay, ionic bonds, they form between positively charged and negatively charged. So positively charged in your amino acid is actually your amine group. Okay, after that your chemistry you will learn that acid structure. You got three types of acid structure proposed by your scientists like Bronsted, Lowry, and a lot more lah. Okay, you got related about proton donor, proton acceptor. So base is actually proton acceptor. Okay, while acid is your proton donor. Okay, so this is actually our ionic bonds that form between positively charged, which is NH3 plus, and also your carboxylic acid COO minus. So ionic bonds are stronger than your hydrogen bond. And the bonds can be broken by pH change lah. Why? Because positively charged is actually can be broken down by very high pH. Because high pH they will produce a lot of OH minus. So all the OH minus they will go neutralize the positive charge. Okay, and vice versa lah. Okay, so this is the second bond that we are talking about. Okay, after that, third part, we got hydrogen bond. So hydrogen bond, they form between strongly polar R groups, and these are the weakest bond that can be formed, but the most common as they form between a wide variety of R groups. So to be simple, hydrogen bonds, they only form between strongly polar group. Polar group means that I can form charge. Okay, I can form charge. And just remember like hydrogen bond only form HF, HO, and HN. These three. Okay, most common one. So hydrogen bonds is for polar group. So hydrophobic charge or hydrophobic interaction is for non-polar. Okay, means that they cannot form charge. Like example, CH2, CH3, those hydrocarbon groups, they cannot form a charge. Okay, so this is the fourth one. Okay, so far everything okay? Okay, yeah, so if this part okay,
Yeah, okay.
okay, we take a short break. Okay, and later we will enter the new things that your IGCSE didn't teach one. We call it globular and fibrous protein. So 390, we continue.
Okay.
Okay, hello. Okay, I think right now we can refresh the meeting. Okay.
Okay.